Look at a single E. coli bacterium. It is a highly dynamic, completely autonomous living organism, navigating its microscopic world. But its physical boundaries are strictly constrained. The entire cell is roughly 2 micrometers long and just 1 micrometer wide. Inside that tiny volume, the cell has to store its complete operating manual, a single circular double-stranded DNA molecule. If you took that circular chromosome, broke it open, and stretched it out, it would measure 1,300 micrometers long. That is enough physical space to encode roughly 4.7 million base pairs of genetic data. This diagram shows the scale of the problem. A 2 micrometer container sits right next to a 1,300 micrometer thread of DNA. You cannot simply stuff a thread that long into a container that small. Without precise architectural organization, the genetic material would immediately tangle into a fatal knot. There is a common misconception that because bacteria lack a nucleus, their DNA simply floats freely in the cytoplasm, like a loose pile of spaghetti. The biological reality is much more structured. All of that DNA is concentrated into a highly organized, dense zone within the cell known as the nucleoid. Unlike the nucleus of a plant or animal cell, this nucleoid region is not wrapped in a protective membrane. It sits directly exposed to the surrounding cellular fluid. We can see this structure in electron microscopy. This image displays the distinct dark inner mass of the E. coli cell. The genetic material is tightly gathered in the center, completely contradicting the idea of a uniformly scattered genome. To achieve this dense centralization, the bacteria relies on 3 specific structural components: a proteinaceous scaffold, a series of looped domains, and DNA supercoiling. By combining these 3 elements, the nucleoid packs the genetic code into a fraction of its original volume, while keeping every gene accessible for transcription. To understand how this works, we start from the base state, a completely naked, uncoiled ring of DNA. The cell first secures the massive chromosome by permanently anchoring it to a single point on its inner plasma membrane. Human cells use histone proteins to spool up their DNA like beads on a string. Prokaryotic cells lack these proteins entirely. Instead, bacteria deploy a different set of specialized packaging proteins, specifically molecules known as HU, IHF, and HNS. This binding relies on electrostatic forces. The DNA's phosphate backbone carries a strong negative charge, and these packaging proteins are positively charged. They snap together on contact. Because the DNA naturally repels itself due to its continuous negative charge, these positively charged proteins act to neutralize that repulsion, providing a stable chemical foundation. While these proteins provide the binding glue, they do not reduce the physical footprint enough on their own. The cell still requires a macro-level physical scaffold. Those individual packaging proteins begin to migrate toward each other, assembling into a dense core at the center of the nucleoid space. As they cluster, they drag the attached DNA inward. The massive circular ring pinches into the center, tethering to the central core at multiple different points. This folding creates a specific geometry. The single ring resolves into approximately 45 independent looped domains, all radiating out from the center in a rosette pattern.
By pulling the outer edges inward to a central hub, the overall diameter of the chromosome drops drastically compared to a single open circle. The rosette structure handles the overall width, but those 45 loops are still relatively bulky. To get this entire complex inside the 2 micrometer limit, the cell deploys one final mechanism of physical compression. That final mechanism is negative supercoiling. In topology, supercoiling involves taking an already coiled structure and twisting it further upon itself to compress it. We can visualize this with a coiled telephone cord. When you twist the cord repeatedly in one direction, it starts punching up, collapsing into a tight, dense series of knots. Looking back at the bacterial chromosome, we can isolate just one of those 45 looped domains. Right now, it's a standard double helix. An enzyme complex called DNA gyrase attaches itself near the base of the loop. Using ATP for energy, the gyrase physically rotates. It forces negative supercoils into the strand, wringing the double helix into an incredibly dense, twisted braid that occupies a fraction of its former length. This process is mandatory for survival. If you introduce a drug like coumarmycin, which specifically inhibits DNA gyrase, the supercoils relax, the nucleoid expands, and the cell dies. The spatial paradox is solved through this strict hierarchy. The electrostatic binding proteins, the looped domains, and the gyrase-driven supercoiling work together to fit 1,300 micrometers of DNA precisely into a 2-micrometer nucleoid volume. If we step back from the nucleoid and examine the rest of the cytoplasm, we find that not all of the cell's genetic material is locked in that central scaffold. Floating independently in the fluid are plasmids. These are very small, extra-chromosomal rings of DNA. Plasmids operate entirely outside the main bacterial chromosome and multiply independently of the host cell's standard division cycle. Rather than housing core operating instructions, plasmids carry modular, specialized tools, traits like heavy metal tolerance or resistance to antibiotics that can be quickly shared between cells. The bacteria separates its vital blueprints from its specialized toolkit. The instructions for basic survival are tightly organized within the nucleoid, while adaptable tools remain highly mobile in the cytoplasm. We can finally view the complete architecture of the E. coli cell, a fully supercoiled nucleoid suspended directly within the cytoplasm. Compacting the DNA is only half the battle. If the cell wants to grow and divide rapidly, that tightly coiled code has to be instantly accessible for transcription. This is exactly why the lack of a nuclear membrane is an advantage. There is no physical barrier between the DNA and the protein-building machinery. As RNA polymerases read the supercoiled loops to produce strands of messenger RNA, those newly minted transcripts are immediately available in the cytoplasm. There is no waiting for transport across a membrane. An army of 20,000 to 30,000 ribosomes surround the edges of the nucleoid. They attach to the messenger RNA to synthesize proteins simultaneously, even while the DNA is still being transcribed. The prokaryotic nucleoid uses electrostatic scaffolds, calculated folding, and tight supercoiling to solve the spatial paradox, keeping the entire genetic code perfectly ordered and instantly ready to support rapid, autonomous life.